Hello, my name is Minnie Liang, and for the SSI Research Bootcamp, my project was done on clustering of single-cell RNA sequencing data via latent space and classification of cancerous cells with marker genes. Pancreatic cancer is the seventh leading cause of cancer deaths worldwide and has the lowest 5-year survival rate of any cancer. 90% of pancreatic cancers are pancreatic ductal adenocarcinoma, also known as PDAC, which is what this project is on. Pancreatic cancer is usually diagnosed at an advanced stage and has very dismal prognosis. On top of that, because of the high heterogeneity of pancreatic tumors, patients benefit very little from current conventional therapy. Hence, there has been a crucial need for new innovative therapies. Recent insight into sequencing data of pancreatic cancer has supported its molecular classification, thus expanding clinical therapeutic options. This study aims to create a machine learning workflow proposed for PDAC sequencing data. This workflow also identifies important marker genes which have prognostic value and potential as molecular therapeutic targets for PDAC treatment. The sequencing dataset used in this study is from a patient's primary pancreatic PDAC cancer tissue and was sequenced by single-cell RNA sequencing technology. The dataset consists of 19,738 genes and 20 different cell types, with 2 of them being cancerous and 18 of them being non-cancerous. Single-cell RNA sequencing is a form of DNA sequencing technology that's able to examine the gene expression of cells at an individual level. And as we can see in this diagram, single-cell RNA-seq is able to look at each single cell type and its expression profile and reveals the cell heterogeneity that in bulk RNA sequencing is masked. We can also look at gene expression in cells. In bulk sequencing, there is no change in expression for gene X, but with single-cell RNA-seq, we're able to look at the expression of gene X in the different cells, and in this case conclude that gene X is affected in cell type B only. In this study, single-cell RNA sequencing is used instead of bulk sequencing, as single-cell RNA-seq is able to explain differing cell-to-cell gene expression that bulk cannot. This study used both cell-level analysis through the clustering of cell types and the classification of cancerous cells, and also gene-level analysis by examining the classification model's top contributing genes. Single-cell RNA-seq has a multitude of promising capabilities, but the drawback is that the data suffers from high complexity. Since we're looking at gene expression for each and every single cell, there's a lot of data and also a lot of zero counts because not every single gene is expressed in every single cell. So this study also aims to address the complexity of single-cell RNA-seq and how to cope with this curse of ultra-high dimensionality and inflation of zero counts. The first part of this project aims to perform cell-level analysis through clustering. First, the single-cell RNA sequencing dataset was reduced to only the top 1,200 highly variable genes to help combat the problem of zero inflation. Then PCA was used to reduce dimensions, and these dimensions were then inputted into UMAP to cluster cell types. This was compared to using the latent space. A package called SSI learns a non-linear embedding of the cells through a neural network, which helps combat the problem of ultra-high dimensionality of single-cell RNA sequencing data. And this latent space with SSI was then compared against PCA to see the differences. So here are the results of PCA versus SSI latent space on the UMAP clustering. And we can see here that the group of ductal cells has higher and better resolution with the SSI latent space than using PCA. So we're able to conclude that the SSI latent space clusters better than PCA. Aside from clustering, marker genes were identified from the cancer clusters in the dataset and used as model gene features to predict if a cell is cancerous or not. When classifying cells as cancerous or not cancerous, the effect of gene feature selection and the effect of downsampling the majority class were also experimented with. Based on differential expression, marker genes were found from the 2 cancer clusters in this dataset. Cancer clone cluster A was found to have 74 genes, and B was found to have 116 genes. 2 settings were tried, either using the union of the 2 sets of marker genes as the model's features or the intersection. And with only 15% of the cell types as cancerous in the dataset, the data also suffer from a huge imbalance. So another 2 settings were tried. 1, not changing anything, or 2, downsampling the majority class. The test dataset was left untouched the whole time, so it was still class imbalanced. So 4 different models were tried out: logistic regression, gradient boost, random forest, and support vector machine. The data was split into 80% for cross-validation training and 20% for testing. It was hyperparameter tuned with GridSearch 5-fold cross-validation with F1 scoring. And before the dataset was actually input into the model, it was first dimension reduced and scaled to a range between 0 and 1 to help combat the problem of ultra-high dimensionality. So here is one of the experiments ran. This is downsampling the majority class versus not changing anything. And this was done by using A intersection B as the model's gene features. And we can see here that the general trend is that downsampling performs better for both the cross-validation and the test set. And this is, if we compare the downsampling and the no change for each of them, we can see this trend, which is circled here. For the test set, we further examined the top 2 scoring models, which were downsampling on logistic regression and downsampling SVM, and we constructed confusion matrices. And we can see with the confusion matrices based on these numbers that the logistic regression model is able to better precisely predict the number of cancerous cell true positives than the support vector machine model. Here is another one of the experiments ran, which is using A union B versus A intersection B as the model's gene features, and this was done on the downsampled data set. And once again, as we compare union versus intersection for each of the 4 different models for both the cross-validation and the test set, we can see that the general trend is that intersection performs better than the union. So here's a table of results for union, intersection, downsampling, and no change, and we can see that the overall best performing model is using A intersection B as the model's gene features, downsampling the data set, and using the logistic regression model. And this was able to achieve an F1 score of 0.85. Based on results, we're able to conclude that using A intersection B as the model's gene features performs better. And this makes sense because the model is learning from genes important to the expression of both cancer clone A and B, whereas using the union could contain genes important to one but not the other. We're also able to conclude that downsampling performs better. And this also makes sense because the model is preventing bias towards the majority class. The next step of the study was doing gene-level analysis by examining the classification model's top contributing genes. Since we first dimension reduced the data with PCA, all the features we're examining are PCA components. The logistic regression model's coefficients can serve as a crude feature importance score. So we can see here that in the logistic regression feature importance graph, PC1, 2, and 3 have the largest absolute value coefficients, so they have the most importance to the model when predicting label. This makes sense that PC1, 2, and 3 have the most importance because based on the definition of PCA, the first principal component always explains the most variability. With each increasing principal component, less and less variability of the data set is explained. A violin plot of feature importance scores from the random forest model was also graphed. And we can see here once again that the first 3 principal components have the highest feature importance. It is also interesting to note that PC 11 and 12 stand out as important features for the random forest model. So with PC 1, 2, 3, and also PC 11 and 12 identified, then the top contributing genes from each of these principal components was found. Then with the SSI UMAP clustering graph, the SSI UMAP clustering graph was then colored by these different genes. So looking at the colored gene expression, it can be seen that the determined genes all have high expression in the cancer clone A and B clusters, as you can see here by the red circles. The results of this workflow analysis can further the understanding of the RNA in these PDAC tissue cells and the understanding of the underlying cellular mechanisms of pancreatic cancer. The identified model's top contributing genes can also be used for prognosis, and with future research in vitro and vivo has great potential as molecular therapeutic targets. As for future work, I would like to quantify the accuracy of the clustering results instead of just visually comparing the PCA versus the SCVI latent space clustering graphs. I'd also like to experiment with the effects of upsampling the dataset by generating new cancerous cell data and see how that compares with downsampling the dataset. I would like to thank my mentor, Minnie, for constantly helping me throughout this project, and also my lab group for their constant feedback, and the SSI staff for teaching me machine learning these 6 weeks at SSI.